Hinanit, it's lovely to see you again, even though it's the virtual sphere. It's lovely to see you again.
Lovely to see you in review. Excellent.
Beautiful. So I am, like all my guests, I'm always excited to speak to them because I really find that the work that they're doing is so out of the box in the cannabis space. Not the common, researcher that's talking about the same thing that they had been researching 10 years ago, but you guys at the Volcani are doing some pretty cutting edge research. So I figure the best way to start is for you to give me an update as to what you're currently looking at in your lab.
So indeed, what we do is we ask questions about cannabis that I believe that almost nobody else is asking. What we ask is what compounds and what combination of compounds are active towards different medical conditions or medical indications. The basic hypothesis that drives our research is that one, it's just a thing of THC and CBD, but rather we are looking at a plant that produces hundreds of different compounds of biological activity. So that's one. The two is that a combination enough to.
Sorry, Hinanit, sorry to interrupt you. Are you able to speak closer to your microphone? Because I think it's cutting out a little bit, but maybe if you speak a little closer to the mic, it will pick you up.
Okay. Internet connection. Can you hear me?
Yeah.
You can hear me. Adam?
I can hear you.
Because, all right. So that we are asking about the combination compounds from cannabis and their activity. For that, what we do is we establish different biological assays in my lab with physicians, with MDs from hospitals, and we examine different medical conditions. By that, we're able to identify cannabis compounds that are active and really have new formulations from cannabis. Listen, we are changing the way I believe that cannabis is being perceived, because no more THC versus CBD or just one medicine to treat all, but rather specific medication from cannabis to treat different medical conditions. And this approach leads to much higher efficacy and specificity in treating different indications. Examples for indications are skin diseases, where we issued a patent and going to clinical trials. And even we are working now on inflammation, including lung inflammation, which is associated with COVID-19. So we have great belief, I would say, which is being assessed by scientific tools, but we are great believer in cannabis and cannabis compounds and their ability to treat different medical conditions.
So can you just explain to me briefly about the medical assays that you're developing?
Absolutely. For each medical condition that we would like to examine, then we establish biological assays that are specific to this disease. For example, once we are working on cutaneous T-cell lymphoma, this is something that we did with Medsi Biopharma company. Then we worked on cell lines, human cell lines of the skin, but also on blood cells and biopsies that were obtained from Beilinson Hospital, our collaborators.
Mm.
Then we could examine directly on patients' cells and tissues. We could examine the effect of cannabis compounds, and we were able to come out with a formulation for the treatment of this indication. So this is one example. Other examples include biopsies that we get from colon tissue. From patients that suffer from Crohn's disease or ulcerative colitis, in collaboration with Meir Hospital in Israel and with Plantext company, we were able to identify from cannabis compounds that can reduce colon inflammation. So these are two examples that we have.
So the Volcani Institute itself is an organization that specializes in agricultural research. Are you guys looking at different molecules in combination with cannabinoids and testing the efficacy of these combination products against any specific indications?
Indeed, this is what we do. We look at different compounds from cannabis, but also from other sources, and the different compounds and their effect on different biological processes that are associated with medical conditions. It means that basically, due to our ability to work with, first of all, with cannabis and then with other plant species, we combine the knowledge that we have in botany, in plant physiology, with preclinical studies. I think that here we touch one of our unique expertise in being able to be in two disciplines. One is the plant and the other is the preclinical studies and the effect of the plants on the human body.
What's the average timeline for conducting these studies? So, you know, how does an idea come past your desk, and then what happens? So does a researcher come to you and say, I want to study this? Do you have an idea that you pursue? What can you explain the process of how a project starts and begins, and what the timelines are on average for taking something from ideation to execution, and then from execution to, you know, clinical research?
So usually the projects that we do are based on collaboration with the industry. As you said before, we are a research institute, a governmental research institute, and then through our tech transfer unit, we get in touch with companies which are interested in doing some research in medical cannabis with the clear aim of having products at the end. We do not just do research for the sake of research, but rather for the development of new products from cannabis. So usually companies are contacting us, and then what we do is we establish this collaboration and start immediately doing the research. Since we have quite a number of years now doing research with medicinal plants and cannabis in particular, then we are able to work relatively fast, relatively. It means that within only few months, we are able to get to the formulation of compounds from cannabis or others that are needed for treatment of certain medical conditions based on the biological assays that we do in the lab. The point is that due to the know-how that we already have in the lab, we are able to move fast, and we know how to isolate compounds, how to analyze first all compounds in cannabis, and then we know how to isolate the different compounds and to look for their biological activity.
So the first thing that comes up for me is the consideration around the complexity of the plant. I mean, let's say a company comes and says, we want to target a particular indication. How do you decipher through the countless number of variables? I mean, there's 500 to 700 compounds in the plant. Like, what's the process to decipher the specific or hone in on the specific compounds to understand what actually has efficacy versus what is noise in the field? So how do you go through that process? Because it seems like you've got a lot of considerations at hand that need to be deciphered. So can you talk about that process? Because I find that particularly fascinating, that area.
If indeed, since cannabis produces so many different compounds, when looking for combinations of one compound with the other, it's a lot of combinations to look at. But what we did during the past few years, we developed our expertise and know-how in such a way that we can look at many different combinations at a time, choose the ones that are working, and then again analyze and separate to different pools. So the idea is to work with pools. It means that at the beginning, we work, for example, with the whole extract. And we ask if this whole extract from cannabis, which contains hundreds or dozens of molecules, is it working? If we see that it is working, then we start to separate it to two different fractions of molecules. We ask for the different fractions, which one is working. We are taking those that are working and again fractionate them to sub-fractions, to again smaller pools of molecules. By doing that again and again, we are able to get to the actual combination that are working. But it's important to say that we also confirm the results using clean standard compounds, which might be even synthesized or purified from the plant. But the main idea is to be able to have this certain composition of cannabis compounds, which is the most effective one. Just to say, but in some of the cases, we even see synergistic interaction between the compounds. Not always, but in some cases. This is so interesting because when it says that you take one compound and another, and it's not one is 20. By that, you are able to increase efficacy 20-fold, for example, or 10-fold. And we think that this synergistic interaction between the compounds is the entourage effect that was reported by Professor Meshulam and co-workers years ago.
I find that fascinating because I immediately start thinking about the work that we do at MediPharm, the company that I'm working with, which is an extraction company. We've got a lot of oversight as to what is being used on the back end across the industry for different relations, and there's a pattern of different companies, like a 1-to-1 ratio, for example. You often will take developing a flower with a perfect 1-to-1 ratio of THC to CBD, as an example, is very challenging. So typically what companies are doing is they're blending. So are you finding that in your research you're able to, let's say, fractionate a particular cannabinoid and then fractionate another one and combine them together and achieve the same efficacy from when you actually had it in the initial plant material?
Very interesting questions that have basically two answers. The first answer is that once you know what you need in terms of cannabis compounds, go ahead and build it from different fractions, different strains. It does not matter. It means that you just have to go then to get into a certain combination of compounds. You can build this combination again using different fractions from the same strain, or fractions from different strains, or just blending of different extracts. But this is all not just by guessing. This is because you know what you need for a certain a medical indication. It means that once you know that you need, for example, a 3% of THC and 2% of CBD, but you need also 5% of CBC and 20% of CBG. You can build it from different strains to have this composition. That's one part of the answer. But I want to give you another part. And this relates to the interaction between phytocannabinoids and terpenes. There is quite a debate now in the scientific literature about the importance of terpenes in cannabis. Some say that you don't really need the terpenes; they're only for for smell or something. But for medical activity, you do not need the terpenes. What we did is we assessed this question using scientific tools, and we checked the activity. First of all, we checked the distribution of different terpenes in relation to phytocannabinoids, and we found that this is not random. It means that when we looked at 70 different strains of cannabis, we found it's going with a certain set of terpenes. So there are terpenes that are specific to high CBD strains, and there are terpenes that are specific to high THC strains. It's not random. Now, based on that, what we did is we took the terpenes of CBD and put it with CBD, with and without, and then took the terpenes and put it with and without. And it turned out that terpenes enhancing these phytocannabinoid activity specifically. It means that only the terpenes that are coming with THC in different strains help THC working. And only those terpenes that are in the CBD strains, high CBD strains, are working with CBD. So one, if I'm getting back to your question, yes, if you are working on the whole extract, it is important to join phytocannabinoids and terpenes from the same strain. They are there for a reason, okay? And we can talk a little bit about this reason if you want.
I'd love to.
Yeah.
Because what I see as an evolution in the cannabis industry is combining terpenes with formulations, but based on what you've just shared, it's not as if you can just take myrcene, for example, and add it to a formulation and expect a sedative effect, or beta-caryophyllene for an analgesic effect. You're saying that it needs to be matched specifically to the cannabinoids that are as it's naturally found in the plant. Otherwise, it's somewhat of a redundant activity. That's absolutely fascinating. I'd love to hear more about your research in this area.
We asked ourselves, why is it so? Why do we find certain terpenes with certain phytocannabinoids in specific strains, and it's not random? And on top of that, as we said, the enhanced activity of these terpenes for the certain phytocannabinoids. What we hypothesized is that this and coactivity of terpenes and phytocannabinoids are a result of the evolution that this plant is going through by human. During the process of domestication, human or man used cannabis for ritual, for medical purposes, for leisure. And then in this process, which was thousands of years. We know about 6,000, 7,000 of years of usage of this plant by man. During this process, men bred and selected for cannabis strains that work the best. Men did not know what he's doing, but rather used the need to select for these specific strains that have high activity, for example, high medical activity, high psychoactive activity. So it means that the co-evolution that this plant went with that of men led to the actual strains of cannabis that we see today. That's very interesting.
Yeah, yeah, that is fascinating. So it's evolved as a result of, the evolutionary process of man's kind of picking and choosing, so to speak, of the specific strains that are best suited for the particular task at hand, so to speak. But it's interesting because all across the world, you've got different use cases of why cannabis be, used as a medicine versus, as you said, for leisure, as you said, for ritualistic purposes. Have you picked up on correlations around what terpenes have been more predominant in different parts of the world or in different parts of Israel? Is there anything that you can share about those correlations between terpenes and location?
Yes, this described we did with the Israeli Gene Bank. The Israeli Gene Bank, during the last few years, imported cannabis seeds from different parts of the world. It imported cannabis seeds from different collections of other gene banks from all over the world. By that, we were able now to correlate between the actual origin of cannabis and the composition. This is a scientific paper that we are working on now and soon should be published. But basically, we have information about the origin throughout the world and then the composition. And this certainly has to do with the usage of this plant by man as well.
Based on the terpenes, from the terpene profile that you've seen in different plants, could you provide insight to me if something came from Africa and the terpene profile, was predominant in specific terpenes? What does that mean versus it coming from the Middle East? Can you talk about what the terpenes are telling you from different parts of the world?
What I say for now is that the clear correlation is between terpenes and phytocannabinoids. It means that it might be that the actual use or the purpose of the phytocannabinoids and not specifically on the terpenes. I will explain. For example, when cannabis strains were designated by men throughout the history, were designated for the use for ritual, for example, then high THC was in need. Then it turned out that only certain high THC strains had the desired activity. These were selected and turned out that these had a certain sets of terpenes. So the actual selection was based on cannabinoids, and then the terpenes were selected as those that the activity of the certain phytocannabinoid. So definitely, as to answer your question, we can, based on the phytocannabinoid activity, we can see sets of terpenes that are going with certain use.
So what are some of the examples of common terpenes found with, high THC versus high CBD?
This is very interesting questions, but you will have to wait just a little bit
Look,
to
you get
be
that's like
published.
okay. So you're currently doing some. So you're going to be publishing some research on this in the near future. Would this be in human studies, or would this be more related to the biological, like in vivo, in vitro studies that you're conducting? Wait, I can't hear you there, Hinanit. Can you repeat that?
Botanical study. A botanical study of which combine the chemistry analysis of the chemistry of the plant. So we can say this is botanical mostly.
Interesting. Yep. My mind's running wild. I love it. You understand the terpene profiles, you understand the botanical makeup of the plant, the terpenes, the origins. You can then also look at the clinical application. It's very interesting. You're basically building the entire block of the cannabis plant to entourage effect. That's really I'm so interested to learn more. So I have to ask Hinanit, if you can give me three, what are the three areas that you're most excited about in your lab right now?
One area that we are so much excited and even, I would say, feel a duty to work on is COVID-19. Since we have a lot of findings and a lot of experience working on colon inflammation, then what we did is we translated this knowledge to working on COVID-19. And we are so excited about research and exciting and unexpected results that we have for cannabis in relation to this disease. Again, I can't say much as for now, but very soon, we are going to go and publish also even immediately as a prep for the public domain our results and new activities of cannabis on the human cells. So this is one research that quite a number of people in my lab are working on, and we're very excited about that. So this is something to come.
Yeah, right.
Another research that we have just completed, and again, this is under peer review now, scientific peer review, is research on tumors that are developing in the urothelial system. We are working on urothelial carcinoma, and then we ask, first of all, is cannabis able to have anti-cancer effect on this type of cancer, which unfortunately repeats again and again in the patients that are suffering from this disease. And then we ask very interesting question, I believe, about the ability of specific cannabis compounds. Remember, always ask about specific combinations, about the specific combination of cannabis compounds in inducing apoptosis in cells, which is programmed cell death, in changing the cancer cell cell cycle. And then we ask about the ability of cannabis in inhibiting the migration of these cells and formation of metastasis. So this is another very exciting research that I do in collaboration with Sheba Hospital in Israel.
Can I just want to ask about your research in cancer, because it's a very broad term. There are many mutations of cancer. What are the commonalities that you're finding in your research around THC's ability to promote apoptosis, or mitigating the cell's ability to receive mitochondria? Like, is it is it just THC, or are there other cannabinoid or terpenoids that are specific to particular cancers, or is there a common trait? Can you talk about this for a moment?
Okay. What we found that different types of cancer needs to be treated at different combinations of compounds. It means that the specific combination of compounds is highly important to have the synergistic interaction. Remember, we talked about the synergy and about the synergistic interactions between the compounds. So what we defined are range of concentrations of relations between the different compounds to have the highest possible anti-cancer activity. So from the plant side, I can tell you that different cancer types needs different treatment, okay, based on composition of compounds. But as you know, different types of cancer are indeed different types of disease, and they're working via different signaling or biological pathways. And then with the specific combination of compounds, we are trying to target the different pathways in the different types of cancer. So in one sentence to answer you, it's not an easy answer. Each and every disease demands its own solution, its own treatment, and the important thing is targeting the actual biological pathways that can be targeted in this certain kind of cancer to have anti-cancer effect.
But is THC the common thread in these approaches, or are there instances THC not even needed?
You cannot say that THC is the common thread, not at all. In some cases, it is needed. In some cases, it is needed only in minute amount. In some cases, it is not needed at all. So I cannot say that THC is the common thread.
That's fascinating. That I find particularly interesting because, you know, the research that I've read in around THC and its use in cancer therapies is it's still quite an uncertain area, and a lot of people will, in at least anecdotal cases, reference THC as the leading agent for their symptom relief for cancer. And what I'm trying to wrap my head around is there that common thread, which it's interesting to hear that it's not. So then the question is, it must be based on the particular cancer type and the particular combinations, and that makes sense, and it does confirm my hypotheses on this topic, but it's not something that I was completely sure of. So you've now closed that chapter, so to speak. But what I want to understand then is further to the conversation around the research that you're doing in the field of cancer. Is there any other indications that you're seeing something like THC being a common thread, or in all the research you're doing, it's completely random in the sense that there are no commonalities? It is always different case by case per indication.
Scientists that are doing scientific work, we do not want to be biased by any predetermination. Because of that, for each medical indication that we work on, we start from scratch. It means that we look at all compounds in cannabis, and as I said, the different pools, and we do not assume activity of any specific compound, but rather let the result tell us what is working. But that we are able basically to get into a combinations that include phytocannabinoids that are rare or when were unnoticed. And by that, we can find really interesting new biological activities for these certain phytocannabinoids and terpenes, as we said, in accordance, just by not assuming or pre-assume assumptions before starting, but rather letting the plant tell us.
Yeah, because my assumption would have been, THC is predominant in the strains that are available. It's the common thread, but it's not the common thread when you dig down and go granular and look at the specifics. So can you talk? I asked you about what you were excited about from a clinical research perspective, and perhaps we'll sidestep there in a moment. But what about the cannabinoids? I mean, as you mentioned, it's usually THC and CBD that get the spotlight, but what are the other cannabinoids that you're currently fascinated by, that you're seeing positive results for treating various indications? Can you talk about a broader range of cannabinoids for a moment?
But just before saying, more specifically, I have to say that once we are talking about the phytocannabinoids, then we are talking about combinations. So it means that if I'm going to talk about a CBG, for example, then it's not CBG on its own, but rather it's a certain combination of CBG with other phytocannabinoids that gives the best activity. So, as said, CBG is definitely a very interesting phytocannabinoid once it is combined with others, and we see clear activities of these phytocannabinoids, again in combination, which is it's really fascinating. We see activity for CBN and for CBC, and also very interesting for the varying varieties like THCV. All of these, again, I'm saying, are not stars on their own, but rather they are working together as an assemblage for different purposes, affecting different biological pathways, having different anti-cancer effects, anti-inflammatory effects, and so on.
So can we talk about THCV for a moment? What I know that it's a combination effect, and if you want to talk about the combinations that you're seeing, by all means, I'd love to hear that. But I could also appreciate that you'll be releasing that via your research papers. But can you talk about the indications that you're seeing? CBC, THCV, and CBN. Where the target, like, what are the indications that they're currently being used in your lab to treat? If you can start by with THCV and then maybe CBN and CBC to follow, I'd love to hear about what these combination of cannabinoids are actually doing to treat different indications, to have an efficacy on different symptoms.
I hope you would excuse me that I can't expose the actual combinations we're working on.
Of course.
But I would be able to talk about information that is already published. And regarding that, in a project that we have with MedC Biopharma, we were, as I said, we identified the combination of phytocannabinoids that are working as anti-cancer against cancer, skin cancer, which is based on lymphocytes, malignant lymphocytes in the skin. This is mycosis fungoides, and CTCL is the disease. And there we found that a very certain combination of CBG with CBD, with just minute amounts of THC, are excellent, very good to kill these malignant cells. And the whole information is published. We published that this year in OncoTarget, and there you can find the actual composition of compounds. What we saw is that this certain composition in the certain ratios is the one who have an effect on the malignant cells, and it is much less able to target and kill normal cells. This was really exciting findings, because perhaps we can target more specifically the malignant cells, have anti-cancer effect with minimal damage to the normal ones. So again, this is something we published in OncoTarget, and you are very much welcomed to look, and we have there all the data and the compositions. As for other combinations, it will come soon, I promise.
Okay, I find myself eagerly awaiting the day that I can read more about these combinations. Let's talk about growing for a moment, because the Volcani Institute, being an agricultural research center, would mean that you are not only able to decipher the entourage effect in the lab, but then how does that play back into the growing of these products? What type of research are you doing in the agriculture space to develop a consistent strain that produces the profile that is used to treat the indication? Can you talk about this for a moment?
I myself, I'm not a breeder, but what we do is we screen in my lab different strains that we have as offsprings of a vast variety of strains that we have, for example, from the Israeli Gene Bank. These offsprings are crosses of, as I said, of a vast variety of strains and from different genetics. Based on that, we have very interesting combination of cannabis compounds, and we are able now to screen for strains that are working the best based on pre-knowledge of the acting phytocannabinoids. By that, we are able to define strains that might be specific to a certain activity. So that's one. One is screening of a vast variety of offsprings of strains. This is one. Another activity of other scientists here in Volcani is actually breeding. These scientists are breeders, and what they do is they breed cannabis for different properties. This might be also pest resistance, high yield, and also certain combination of compounds for different medical indications. But they're based on information that we provide, but the breeders can do breeding in a rational way to have offsprings with desired properties, desired traits. So definitely this is an activity that is ongoing in Volcani.
So with the cannabis compounds that you discover, because you mentioned before you discover minute cannabinoids that have a very important role in the treatment or in the targeting of particular indications. How do you at scale then provide these compounds to develop these formulas? Are you then going back to the agriculture center and saying, we need more of this cannabinoid, you need to now develop a breeding program for this? Or do you move to a synthetic model? Or is it a combination of both?
Indeed, for taking the results of the research and moving towards a new cannabis-based drug. What we do is, as I said, we collaborate with the different commercial companies. The companies are the ones responsible for the development of this product. The approach that we take is that we translate results that we have in the lab to commercial activity by working with the companies in developing the pipeline of product production, development and production. It means that then, once we know what is the composition based on the research, then we go with the company, let the company upscale the extraction of certain cannabis strains. We work with them to see the composition, to see the biological activity, and then the companies take these certain compounds from cannabis and put them into a certain delivery, drug delivery method or formulation and so on. So it means that the approach that we take is based on cannabis, first of all, and usually not on synthetic compounds, one. And two is that with the companies, we build the pipeline of production of the product, based again on the knowledge that we gained in the lab. By that, we're able, for example, to take different fractions, as we said before in the beginning of our conversation, different extract, we blend, we mix, we check for composition, for activity. By that, we're able to get cannabis-based products. It can be done from existing strains. To be honest, we don't have to go to breeding and wait for the breeding strains to be established. However, we can take from strains, from commercial strains, commercial genetics that we have today, and build the new medical-based drugs.
And what about extraction? I presume you've got multiple modalities for extraction. Do you take the same strain and extract it with different extractors and then test based on the different extraction methodologies?
What we did at the beginning, a few years ago, we checked different extraction methodologies. For us, the most important thing is to get a vast array of molecules as much as possible. For now, we use mainly two methods of extraction. One is methanol-based extraction, ethanol-based extraction, and then dissolving in methanol. But for the commercial use, it's definitely ethanol-based. That's one. And the second one is hexane-based. Hexane might be more problematic for commercial production, but this is a very important tool for us. So for commercial production, we definitely suggest to use ethanolic extracts. That's one. Two is the separation to fractions. To be able to work with companies for production, what we practice here is a flash chromatography, which allows us, using this flash chromatography, to divide the extract to fractions. We chose this because the industry can use these kinds of apparatus, just upscaling, and by that we can move the research results into the hands of industry as quickly as possible, as easily as possible.
Okay, yeah, because that's the tech transfer piece would be challenging, especially considering that, you know, a lot of these facilities would then need to validate those methods in-house and basically test the methods, ensure that they're covered by their relevant license, and then if they're going to export them, ensure that they're you know, GMP certified for production. So there's a lot of it's such a complicated process. just by what you're sharing with me, my mind's going in multiple directions, and I want to translate that or look at now how your work's being currently used to help patients in Israel. Are there any current trials that you have right now in Israel that are being used in patients? And can you talk about these different trials?
Some trials I cannot talk about yet because they are at the very early stages. One clinical trial that I can more freely talk about, again, because this was published, because the results of the preclinical studies were published, as I said, in OncoTarget. This is the project of treating a skin cancer, as I said.
Yeah.
And here with Beilinson, we are going into clinical trials with products that were already established by certain by certain company. And this is going to be very interesting, very, I would say, a very exciting clinical trial in trying to use cannabis compounds as anti-cancer drug, not for palliative care, but as anti-cancer.
Anti-cancer. Yeah,
So this
wow.
is definitely something we are very excited.
I've been informed recently that CBD in Israel is not popular with patients. First off, how do you see that? And second, is there any research that you are doing now in the lab in patients that are using CBD predominant strains? Because ultimately what I'm hearing is that it's primarily led by, or Israel's primarily led by THC predominant products, but obviously there's a lot of evidence that shows that CBD can be used for a variety of different indications. What are your thoughts on this, and how is there a need for more CBD based products in Israel? Does your lab have any projects that are currently investigating that?
First of all, it is news to me. I didn't know that CBD products are not in Israel. What what I can say on this subject is that Israel is going now through an interesting regulatory process, I would say, where until now, and even now, cannabis is forbidden for recreational use, and not even CBD products are allowed. It means that unlike in many other places in the world, CBD is considered under as a drug just as THC is. This was, and this is the situation even now. However, due to some decisions that were being taken by the regulator and the ministry and the government, we see just the starting of a change where CBD products are going to be considered like in the other world as a non-regulated, at least not as a drug. It might be regulated as a food product or OTC, but not as a drug as it was until now. So this is a less scientific, but more social and regulatory process that we are going through now. There were even some discussions on fully legalized cannabis. I don't know. We'll see how this progress.
Do you think that Israel will fully legalize cannabis in the next 5 years?
I really don't know. I don't know. It's it's quite a question here in Israel. It's quite a debate about social movements that are demanding and would like very much cannabis to be legalized. but many others see this as a not a very positive movement, at least not for us in Israel.
Yeah.
So
Yeah.
sometimes we say, I don't know.
Yeah, that makes sense. I find the topic interesting because ultimately the important aspect is to get the medicine right first, and then bring in the recreational. But at the same time, I'm also very much in favor of adult use cannabis. It's just it needs to be it needs to be considered so that the medicine's put first before anything else. So yeah, I'm interested to understand one final area because we're almost on the hour, and I'm mindful of your time. Back to the terpene discussion. So we explored interesting cannabinoids. Can we take that same line of thinking around terpenes? What are some of the terpenes that you're most fascinated by in the lab?
Yes. As for terpenes, we discussed about their terpenes' ability to facilitate cannabis activity. And as for now, I cannot point, unfortunately, I cannot point to which terpene is the most important one. This is a research we are still conducting. I can talk about groups of terpenes that are working with phytocannabinoids, as said, but I cannot say this terpene and that, but just groups. And
Yeah.
this is something, again, we published in Molecules in 2019. I have to tell you another something, is that, and that is we've done multiple tests in the lab, at least for the indications that we're working on, and we do not think the terpenes are working on their own. So they are very smelly but to say the terpenes have a medical activity, this is something that I find very hard to say. I think, at least from what we see in the lab, is that they are mostly accompanying phytocannabinoids as part of the entourage effect. But I wouldn't say, as for now, that terpenes are working on their own. This is the most, the best that I can tell you as for now.
What about the companies that are looking to, I mean, there will definitely be companies that will be looking to add caryophyllene to their THC product. But the question that then arises is how much would you add, and what other combination terpenes need to be added, and the dosages required for any type of efficacy. Are you doing any research in that specific area? And are you also looking at combining terpenes that are produced from other products? So even though they might be derived from nature, are you taking terpenes that are not derived from cannabis and reconstituting them into new formulations?
Very interesting points that we can discuss. One is the amount. The question is how much to put, how much of the terpenes to put into the different extract to have the best activity. Interestingly, what we found, and again, this is something we published, is that you need to put terpenes about 1/10 only of the amount of phytocannabinoids. And this is the amount that you find in the plant. In approximately, you have about terpenes from the amount of phytocannabinoids in the plant. And this is the active range. And indeed, when I said there is a debate in the scientific literature about the activity of terpenes, in many of the cases, we found that once terpenes were inactive, they were just added in higher dosage to high terpenes, their entourage activity. So that's an important point that we have not discussed before. Another point that you raised is about terpenes from other sources. And here I can share with you something we have not published yet, but definitely I would be happy to share and to say that we have isolated terpenes from other plants. And we were able to see the terpenes from other plants, of course, after we identified them and added them specifically, they act similarly. It means that there is not something unique in cannabis terpenes, and once you find beta-caryophyllene in another plant, then you definitely can use it as if it is beta-caryophyllene from cannabis. And so that's another answer to your question.
Yeah, that's fascinating because yeah, the question is whether or not the other terpenes would work the same or similarly. That's good insight. Thank you for sharing that. And then my last question around terpenes is the consumption of terpenes. Is there a better medium for consumption? Because my concern would be for inhalation, whether it be vaporized or smoked as a joint, whether the terpenes would then degrade because of the heat. Is there a better way to consume terpenes?
I'm not an expert in consumption, so it's not easy for me to answer about consumption ways. What I can tell you is something else that we examined and again published, and this is sort of a more agricultural study. We asked, I think it was published in 2018, I think in industrial crops. And what we looked is at the composition of terpenes in the inflorescence, in the flower, throughout the plant development. And what we found is that even with heating, the terpenes are being degraded from diterpenes and sesquiterpenes into monoterpenes. So it means that the composition of terpenes is changing. Now, if you take into account what we talked before about the importance of terpenes as entourage to the fact change the composition, you change their ability to act as entourage molecules of phytocannabinoids. So definitely, just to say that this is a very important subject that should be considered once delivery methods are being assessed.
Yeah, I mean, well, THC degrades quite quickly. So, do terpenes maintain their integrity through refrigeration? Is it better to store these products in cold chain conditions versus leave room temperature because of the degradation? And do terpenes degrade as, not all of them, it's a broad statement, but in your research, are you seeing terpenes degrade as quickly as THC, or how do they maintain themselves as a molecule? CBD is quite stable, I've seen from the research, but what about terpenes?
Something which is out of my expertise, but what I can tell you is that from what we saw, is that terpenes are easily being degraded, easier than the phytocannabinoids. So definitely this is something to consider. Easily, they have been easily degraded from, as I said, from sesquiterpenes and diterpenes to monoterpenes, and again, something to consider.
I'd love to see the research in terpene integrity and how to maintain the terpene profile. It's definitely an interesting research project.
An important one, so important because of the different products that you find on the shelves. So determining shelf life.
Are you doing that research?
No, we are not doing research in terms of for shelf life.
Okay. Hinanit, I could keep going. I appreciate that you've now given me more than an hour, so thank you so much. It's definitely a to be continued because there are more questions, but it's an absolute pleasure to speak with you, and I'm truly grateful that you could give me this time. Thank you so much. where would be the best place for people to find your research?
I think that, first of all, in the scientific literature, once you go into Google Scholar and look for my name and cannabis, you can find our scientific publications. Some of them are scientific papers on research, but some of them are scientific papers as reviews and opinion. These are, I believe, quite easily approachable and are able to better present in more details our activity. And people are very much welcomed to contact me, and I would be happy to send them PDFs, copies of our publications. So, and to answer any questions, of course.
Awesome. Hinanit, thank you so much. It was an absolute pleasure. And I'll speak to you again very soon, I'm sure.
Thank you so much. It was a pleasure to me as well.
Thank you, Hinanit. Always indeed. Many more to come in the years ahead of us. So take care and all the best, Hinanit.
Thank you. You too. Bye-bye.
Bye-bye.